All right, so the next part of this is going to be looking at, what happens when we take a look at more than one trait at one time. So up until now, we have pretty much dealt with Punnett squares, working with one trait at a time. So what happens if you have tall versus short, or green versus yellow, or whatever? But what if you have to deal with both tall and short and green and yellow at the same time, where you have both color and height? So that's kind of what we're going to talk about today. So the question is, how do alleles segregate when more than one gene is involved? The principle of independent assortment states that genes for different traits can segregate independently during the formation of gametes. So they do things separately. So Mendel wondered if the segregation of one pair of alleles affects another pair. Mendel performed an experiment that followed two different genes as they passed from one generation to the next. Because it involves two different genes, Mendel's experiment is known as a two-factor or dihybrid cross, di meaning two. Single gene crosses are monohybrid crosses, so that's what you guys have done so far. So here's what this will kind of look like. So Mendel crossed true breeding plants that produced only round yellow peas with plants that produced wrinkled green peas. So you can see here we have individuals that are homozygous dominant for both, okay, over here, and homozygous recessive for both here. So this is very simple, very straightforward. They're all going to be the same because they're all going to get the same combination. But what I do want to show you here is, because this is the first time you're seeing this, is so now what you're going to put here is, so this is one parent and this is the other parent. You're going to put every possible combination of R's and Y's that you can put in here, giving one R and one Y to each. So basically the way you do this is just like in algebra. I'm sure you guys remember the FOIL method, first, outer, inner, last. You do the same thing. So if you take the first of each, that's big R, big Y. Then the outer of each is also big R, big Y. Then the inner of each is still big R, big Y. And then the last would be this R and this Y. So these are all the possible combinations of alleles that you can get. Up here, same thing. These are all the possible combinations. This is the first, outer, inner, last, right across the top there. So that's how that's going to work. So the round yellow peas had the genotype big R, big Y, which is homozygous dominant. Already went through that. There's the homozygous recessive for both on the other side. All of the F1 offspring produced round yellow peas. These results show that the alleles for yellow and round peas are dominant over the alleles for green and wrinkled peas. The Punnett square shows that the genotype of each F1 offspring was big R, big Y, little y. So it's heterozygous for both seed shape and seed color. So they would all be exactly the same. Mendel then crossed the F1 plants to produce the F2. So he took all these guys in here and then crossed them. And Mendel observed that 315 of the F2 seeds were round and yellow, while another 32 were wrinkled and green, the two parental phenotypes. But 209 seeds had combinations of phenotypes and therefore combinations of alleles that were not found in either parent. So I'll show you kind of what this looks like. Let's see. Oh, they're not going to show you in here. Okay. So I'm going to go through a problem like this with you guys later where it actually shows. But one of the important things here, Mendel's experiment results were very close to the 9:3 to 3:1 ratio that the Punnett square predicts. And we'll see that later. Mendel had discovered the principle of independent assortment. The principle of independent assortment states that genes for different traits can segregate independently during gamete formation. So summary of Mendel's principles. He basically figured out heredity, observed through patterns of inheritance, form the basis of modern genetics. So this is all the stuff that we already talked about. We're going to kind of skip through here. And then we're going to get to stuff that is different than what we've talked about so far. So there are some different scenarios that you can possibly have. Mendel's principle offers a set of rules which lead to very predictable patterns of inheritance. There are exceptions to every rule and exceptions to the exceptions. So what happens if one allele is not completely dominant to the other? And this happens a lot. So far what we've been dealing with are very, very genetically simple things, but not everything works that way. So what are some of the exceptions to Mendel's principles? They are either some alleles are neither dominant nor recessive. Many genes exist in several different forms and are therefore said to have multiple alleles. And we're going to talk about all these. Many traits are produced by the interaction of several genes. So despite the importance of Mendel's work, there are important exceptions to most of his principles. In most organisms, genetics is more complicated because the majority of genes have more than 2 alleles. In addition, many important traits are controlled by more than one gene. Mendel's principles alone cannot predict traits that are controlled by multiple alleles or multiple genes. So this is an example of what we call incomplete dominance. So here we have a red flower, and that's big R, and we have a white flower, which is big W, but neither red or white dominate the other one. So what you get here is a blend of the two. So all the offspring in here would be capital R, capital W, which would make them all pink. So you get a blend of the two. So that's what incomplete dominance gives you, is a blend of whatever your two traits are. Codominance. Cases in which phenotypes produced by both alleles are clearly expressed are called codominance. For example, in certain varieties of chicken, some of them called erminette chickens, the allele for black feathers is codominant with the allele for white feathers. So what you get is chickens that have both white and black feathers together. It's not gray, it's black and white. Another different example are multiple alleles. So you guys, I'm sure, have heard that there are different blood types. There's A, there's O, there's AB, there's, O positive, O negative, B positive, B negative, A, all of these different ones that are over here on the pie chart. So these are said to have multiple alleles. So many genes have multiple alleles, including the human genes for blood type. This chart shows the percentage of the US population that shares each blood group. Traits controlled by 2 or more genes are said to be polygenic traits. Polygenic basically means many genes. Polygenic traits often show a wide range of phenotypes. The variety of skin color in humans comes about partly because more than 4 different genes control this trait. So that's why we all have different skin tones. I'm going to go back to this one just for a second, and then we'll end here. And maybe I'll give you an example. I'm actually going to write myself a note to give you an example of this one later and show you kind of how this one works. But the polygenic traits, imagine this. Imagine you have a different gene that codes for how long your legs are, and then how long your torso is, and then how long your neck is, and then how long your head is. Well, all of those genes together will determine how tall you are. Okay, it's not any one of them individually. It's not like there's just one gene that says height. It's that you have different lengths of different bones that all add up. So that would be an example of a polygenic trait. Okay, so I'm going to stop this for now, and then I'm also going to give you guys a whiteboard talk where we go through a dihybrid cross and the blood typing situation.